Okay, hello, welcome. Welcome to today's webinar, Johnson & Johnson, a hybrid SAS R submission story, brought to you by the R Consortium. The R Consortium supports key organizations developing R infrastructure through grants and sponsorships worldwide. Please visit our website to learn all the details and how your organization can become a member. My name is Jesse Kasman, today's moderator. We consider webinars an extension of the community. Please feel free to ask questions. You can type in any questions you have through the Q&A icon at the bottom of your screen. At the end of the presentation, the speakers will be responding. Ning Leng is on the webinar today to help handle the Q&A session. Ning is the interim global head of the Data Science Acceleration Group at Roche. The R Consortium hosts free educational events on R-related topics regularly. Coming up in 3 weeks, November 12th through 13th, we are hosting a 2-day fully virtual event on R plus AI. We have an incredible lineup of speakers. You can get past the AI hype and find out if and how you can use AI with your R programming needs. Please visit our website to find out more. Okay, let me very quickly introduce our 3 speakers. We have Stephen Hasendonks, Statistical Programming Lead at Johnson & Johnson. Alicia Humphreys, Associate Director at Johnson & Johnson. And Linshan Yuan, Statistical Programmer at Johnson & Johnson. Okay, I think we've already got the slides up. And Alicia, I think we're starting with you.
All right. Thank you, everyone, for joining our presentation on our hybrid submission strategy. And just to highlight some of the items that we're going to go over today in this presentation is a quick discussion on how we identified the trials for the hybrid R SAS submission. And then additionally, some information around the validation of packages within our statistical computing environment and our overall hybrid R SAS strategy. We'll talk specifically about 2 successful submissions to the FDA, one of which is already approved and the other one has had a positive agency response, and end this with some solutions and challenges going forward. So the content of this presentation, we're going to start with the pilot trial selection and then move into the in-house developed packages to open source packages, discuss the ADRG with the appendices that includes the R environment setup. We'll walk through the R TFL development to hybrid SAS R submission. We'll talk high level with interactions that we had with the health authority agencies and end again with those conclusions and challenges going forward. So our pilot trial selection, we started with an overall approach on identifying early adopters, just adopting R as a programming language for our outputs. So our tables, listings, and figures. We identified newly starting trials from different therapeutic areas and in different phases, but specifically looked for those that had submission potential. Of those, there were a few that we earmarked as potential submission pilot trials. And from that, there were 2 that we followed through from 2 different therapeutic areas that successfully submitted to FDA that we'll be speaking about today. So a little bit of information around our hybrid SAS R strategy. In this, the ADAM datasets were still created using SAS. And the reason for that was because we have a large SAS macro library, a lot of standards, and then just the basic knowledge around each of the disease areas. So we decided to keep, for these 2 trials, the ADAM datasets in SAS. But the TFLs were generated in R. We found it to be more flexible and especially a lot better visualization of graphics. And we benefited a lot from the open source community. Within this, we were able to use some of the cutting edge statistical methods, and we seamlessly moved our workflow integration from a primary language of SAS for everything into a multiple language type environment.
Before we dive into our submission strategy, it is important to highlight two in-house developed R packages that are currently open sourced. The first one is called MV Setup, and it is managing the input and the output locations of a file during its life cycle. By that, I mean that a typical program would start in the development area, after which it would migrate to an area where QC can occur. Then at the end, it would move to a production environment, a production location, from which deliverables can be distributed to internal and external stakeholders. It is quite tedious to manage those input and output locations in a static way, and therefore we had developed a package that allows us to control those input and those output locations in a very dynamic way throughout the entire life cycle of a program, depending on where that program is currently residing. All of this is happening through a small configuration file, which is specifically tailored to our statistical computing environment at Johnson & Johnson. And this is already the first challenge that we had to overcome, because this needs to be transferred to the reviewer, and they are working on a different system. So that configuration file had to be retailored again. And we'll come back to that later down in this presentation. Three key functions of this package. The first one is our profile. And what it is doing, it is reading in that configuration file and then making all those dynamic paths available to the user. Then we have a function that allows to read input files from such a dynamic path. Like, for example, here we are catching ADSL from an input location A in. On the other hand, we should also be allowed to write to a dynamic path, and therefore we have the function write path. Next slide, please. The second package is called tidyTLG, also open source, and this package allows us to summarize data in tables, figures, and listing using the tidyverse as a backend. Again, key functions are summarized on this slide, and we have categorized them a little bit depending on their functionalities. The first general descriptive functions allow us to summarize univariate and categorical data. Like, for example, univar we use for univariate statistics. We have a function called TLG setup, which allows to add column metadata to the summary. And what it is actually doing is making sure that the right statistic ends up in the correct treatment column at the end when we generate the output using GenTLG. Then we also have one small utility function that we wanted to highlight. It's called round SAS, and as the name implies, it's doing a rounding of numeric values as we are used to seeing as it has been done in SAS. Next slide, please. In this slide, you see how those two packages nicely come together in a typical script that we have used for the submission. And you see that the scripts are very nicely organized. So we first start with calling the necessary libraries. We define some variables that are used throughout the script. And then we use MV Setup to read in ADSL from the dynamic path A in. We're doing some filtering. Next, we do summaries using a univar function from tidyTLG. And we store it in a table called T2. Then we are combining all of the summary tables that we have created during the program. We apply column metadata to make sure the correct statistic ends up in the correct treatment column. And then finally, we are going to output that final table to an RTF using the GenTLG function. Next slide, please. In the next section, we want to focus on the Analysis Data Reviewer's Guide, or the ADRG, because it's a critical document. It really talks about how the analysis is set up, through how to set up an analysis environment, all the way to explaining how to run the programs. And don't worry, we will not go into every little detail of the ADRG. We just want to highlight two specific parts that were important for us for the hybrid submission. The first part is section 7, which will be covered on this slide. And the next part is appendix 1, which we will cover in the next few slides. The ADRG that we have used in this submission is modeled after the one that has been published by the R Consortium during the pilot 3. In section 7, on the left side, you see red boxes around some of those smaller subsections, and that's where we made some minor modifications. And all of the modifications that we have used were due to technical limitations or due to specific submission requirements. In 7.3, you see that we list out all the R programs used, and we also include all the functions that were captured in those programs. And we have chosen to prefix every function inside a program with the package for full transparency. In 7.5, we explain the packages that we have used that might not be very familiar to the reviewer. And in 7.6, we include some commonly R functions that are playing an important role in our scripts. Next slide, please. So in the next few slides, we will cover the appendix 1, which we also copied from the pilot 3 ADRG from the R Consortium. On the left side, you see the one from pilot 3. On the right side, you see the one that we have used. The content is almost exactly the same. We have just chosen to be a little bit more granular and included a few more titles to make it easier to follow all of the steps that are required. The first step, installation of R and R Studio, we will not cover. We will immediately skip to step number 2, where we are defining the working directory. Next slide, please. So in the ADRG, we ask the reviewer to create a specific folder structure on their C drive, and that has to do with the MV setup package. And I'll come back to that in a few more slides. And the specific folder structure contains a few folders. So the first one is an add-on folder in which we ask the reviewer to put the SAS datasets after they have been converted from the original XPT files that were submitted. In the CodeLib folder, we ask them to place all the R functions that are available in the submission. Now at Johnson & Johnson, we have the convention to convert all of the programs, functions, macros, whatever, to a TXT file during the submission. Now, in a hybrid submission where you are submitting SAS and R programs, it is quite confusing to understand which files need to be converted back to an R or back to a SAS extension. And therefore, we have made a decision to, before we convert the files, to include the original extension of those files to the file name. So hence, that's why you see, for example, here -r.txt, which we have submitted, they need to be converted back to the original extension R. The same for the programs folder. There we put the programs. We have an input folder where we place metadata files, and then we also ask to create an output folder so that the reviewer can output the results of the programs. Next slide, please. So in this slide, we try to make it a little bit more digestible, what we just said with a visual representation. So here again, you see on the left side, the XPT files that need to be converted and placed inside the add-on folder. Then we see functions and programs that need to be moved to the corresponding CodeLib or the programs folder. And you see on the left side that -r.txt, so that's the extension that we submit, and we ask them to convert them to the original R extension. To facilitate, the splitting between what's a function and what's a program, we provide additional table of content PDFs for the reviewer. And then in the MISC folder, we have some input files that need to go to the input folder on the right side. Once everything has been moved around, we also ask the reviewer to create an R project of that folder so it's more easily managed later down the road. Next slide, please. Then once the folder structure has been set up, the reviewer is required to start installing the packages. And it starts with a remote package because it allows them to install packages from remote locations such as CRAN and GitHub. And another package we ask to install is the renv package because this supports a reproducible environment. It locks it down after it has been created and allows reviewers to come back time and time again to reproduce the exact same analysis. Next slide, please. So the next step is to install the packages that were used in the analysis. And this is where we are slightly deviating from the approach used in Pilot 3, where they proposed to use a renv log file that captured that exact information. However, we experienced some technical difficulties with making that file transferable between a Linux system, operating system that we have at Johnson & Johnson, and a Windows system which is used at the reviewer's end. So to resolve that technical issue that we experienced, we provided a small configuration file inside the submission package that contained exactly the same information, including the package version, the source that it needed to be downloaded from, and the package name. Next slide, please. Once those packages have been installed, we ask the reviewers to run a specific piece of code to lock down that environment so that they can easily reproduce the analysis when they come back to the project. Next slide, please. Okay. So now the folder structure has been set up. The reviewer has all the packages installed. The only piece that is missing is the input and the output location. So remember when we first started discussing this, Johnson & Johnson, we have a dynamic way of assigning paths, and we need to translate that now to the more flatter structure that we proposed for the reviewer. To facilitate this, we also provide that configuration file and we tailor it to the reviewer system. And you see in that TXT file on the right side, you'll immediately recognize those folder names that we proposed. And that's also why we were that strict, so that the MV setup TXT file is more easily transferred and they wouldn't need to make any major modifications. To make those paths available to the reviewer, they need to run the rprofile function. And to make it a bit easier again to the reviewer, we propose to include all those lines of code inside an rprofile, which is made available when they create a project. In addition to that, we also ask to include some lines to automatically source the code library folder so that all the functions are available. The result of that is when they start up the project, in the back end, the rprofile is read, the paths are made available to them, and also the functions are made available to them. And they can just immediately run the analysis and just focus on that. Next slide, please. And then finally, the reviewers are able to run the analysis. And then now I give the word to Ling Shan, who will go into more details about the actual submission.
Okay, so from now on, so let's talk about how this hybrid strategy is implemented when we work on the studies, from the analysis output development to the submission package preparation. So here, this is just a snapshot of how we organize the analysis outputs in two studies. And these two studies have submitted later on. And this is they are also the good example to show how we utilize both the SAS and R in the analysis workflow. So at the very beginning, as shown in the top, all the ADAM datasets are created in SAS. But then almost everything downstream means that all the figures, tables, and the listings were all created in R. As an example, you see all on the left, we have all the subject information, safety analysis, and the lab data analysis, and the PK data analysis, the PD data analysis, even the anti-drug antibody analysis. They all handled in R using the key packages like TidyTLG, dplyr, and the tidyr. And in the middle, we use R for all the graphics. And we list all of the kinds of the plots we are using R to handle in these two studies using the ggplot package. For the primary and the key secondary, as you can see on the left side, there's two scenarios. For one of the study, all of the primary and the key secondary analysis relied on the statistical R packages, such as survival package for survival analysis, EPIR for the binary analysis. Another situation for another study, as you can see on the far right side, is primary and the key secondary required MMR analysis, which is handled in SAS, and we save those MMR result in SAS dataset format, but we're still using R to put to take those result, those value from dataset and display into the RTF files using the dplyr package or TLG packages. In this, all of the R package used here is open source package, including the package TLG. I haven't set up these two packages developed in-house, as Steven explained in the very beginning. Both packages open source package as well. So very important. All the packages used in these studies are open source and also validated following the J&J GSP standards. Based on our experience, it shows that one of the benefits using the open source package could be make the submission more straightforward to work with. Next slide. Okay, so we want to summarize how we conduct the verification for R-based TFLs result. We want to look at this from the three major components. For the general summary tables, we use the SAS standard macros, which proves very stable in many years. And then we use those standard macro to generate the QC outputs. Those outputs could be saved in the RTFL format and then compare against the R-generated RTFLs by extracting their contents either through the R scripts or SAS utilities, from which those provide many options, allows us to conduct a comparison. It makes the QC process more efficient and then more efficient. And then next one, so we want to focus on the critical statistics method. Here we re-implement the analysis using the SAS procedures to cross-check the result from R. If any difference arises, then we were working with the team to investigate the root causes and take the corresponding step. And then the last one is when we QC or verification the graph of figures result, we could utilize the R functions such as like ggplot, the build function, and this function allows to extract the underlying data from the R plots, such as those generated by the ggplot packages, and we can save those. Those extracted value could be saved in the RLog file and used to cross-check the values in the corresponding table, ensure there is a consistency between the figures and the tables. So you can see here the multiple language, we can utilize the strength from multiple language. And here we're using both SAS and R as an example. So this approach allows us to utilize those strengths to move the overall verification more efficient and more robust. Next slide. Okay, so this slide shows the structure of how our ADAM submission package prepared and designed to support this hybrid SAS and R strategy. And for the ADAM dataset folder, all ADAM still saved as XPT format. And when you look at the program folder, we have both the SAS program file and R program file mixed together. Each each script will convert it to the TST, but with the suffix to indicate the original program. And we also create a separate table of contents in PDF files. You can see here we have the individual TOC for SAS program, also the TOC for the SAS micro, also have separate the TOC for the R program, separate TOC for the R functions. So this way could allow the user to navigate easily and then has the much transparent to locate those programs. And then in the miscellaneous folder, besides the metadata file like title Excels, we also include the 2 additional configuration files. These 2 configuration files, as Stephen explained in the previous slides, these 2 configuration files were used to support the R environment setup, support the instruction mentioned in the ADRG appendix. We include this configuration file in the submission package as well. And then also back to the ADRG, we incorporate the updates based on the particular study working with, make sure this section 7 include all the essential R function our package used in the study. And also talking about the appendix, this is the new section added for this hybrid submission. Make sure the overall R environment setup instructions is clear, and we testing those instructions, make sure those following those instructions, the user can be able to reproduce the same result as the RTF script in the Windows system. Next slide.
So now we're going to talk a little bit about the interactions that we had with the regulatory agency, specifically FDA. For both submission 1 and submission 2, as was mentioned earlier, we did propose in the pre-submission briefing book that we would have a hybrid-based submission where they would receive ADAM datasets in XPT, SAS and R scripts both, but in TXT format, the ADRG as a part of the submission package. And then in both submissions, we had asked for a mock submission where we could send the agency a couple dummy ADAM data sets, some representative table and figure scripts in the TXT file, an ADRG with the instructions. So then we could make sure that the agency could follow those steps and prepare their environment appropriately to have that reproducibility on their side. In submission 1, FDA agreed, and we did go through that mock process and submitted that mock through the FDA gateway. Submission 2, which remember was a different therapeutic area, so it's a different review division. They did not want to move forward with a mock process. The response, in a nutshell, had said, as long as that your submission package is following all of the guidelines, then there should not be an issue. And then in our real submission, we did submit all of the ADAM data sets in XPT. And as Linshan had mentioned, for submission 1, we did have that MMRM result data sets out there in XPT as well. For submission 1, we submitted over 200 TLF scripts in R. Submission 2, we had over 300. And remembering that we actually sent the files down in TXT format. And I did see there was a question in the chat, and maybe it's worth just going ahead and answering it right here. So the rationale why we decided to stick with the TXT format, even though FDA does accept now the .R format, was just because our internal J&J processes are set up really more SAS-based at the time that we did these 2 hybrid submissions. So transforming all of the files into TXT format did allow a smoother flow through our internal processes and getting the submission out the door. And then this is just a graphical representation of the 2 submissions. You could see in both, we did have that briefing book where we proposed the hybrid submission plan. and did get the okay from the agency. And then for just the one submission, we did have that mock, and it did work out very well. So what ended up coming out from that mock, we did submit, that small package to FDA. There were some hiccups on their side, especially with a different operating system. And we ended up having a 30-minute informal meeting with FDA where the reviewers shared their screen, and Stephen on the call here, real time, QC'd and got the reviewer to be able to set up their process on their side, so that everything was running. Our takeaway from that is a big lessons learned that we're sharing here in this presentation on how then we ended up setting up the ADRG, how we ended up creating the environment for them in the submission package. And the result, submission 1 has already had FDA approval, and we also prepared a similar package for PMDA, and that has also been approved, submission 1. Submission 2 has received positive feedback from FDA. We have not gotten the submission or the approval yet. So in conclusion, we recommend that you adhere to open source packages when you're able. This helps avoid the extra work in the submission, and it really simplifies the setup process for the reviewers. To be very explicit about the R packages that you are using and ensuring that they follow the GXP compliance. Enhancing readability of the R analysis results. And that involves giving explanations on your programming logic, our session information in the log file, and then having that internal validation. To have your atoms be as close as possible to your TLFs, that old SAS terminology of the one proc away, to help minimize the customized functions to handle any additional derivations and manipulations, just helps with the simplicity of running our scripts. And then ensuring in your ADRG that you are clear with the package information that's included in the R environment setup. And we recommend taking the Consortium FDA pilot projects and other R submission cases as your reference. Additionally, using a single file which contains all your R packages, and include that in the submission package to help simplify the installation process for the reviewer. And the revloc file is required to create after installation, and it serves to stabilize and lock the project environment. So then you meet the requirements of the ECDD file structure and file formats. So some of the challenges and recommendations going forward. So having an internal testing can help ensure that you are replicating the sponsor's environment. They're replicating the sponsor's environment the best that they can. Recognizing that different operating systems require different settings and generate different warnings, and that different R versions don't necessarily perform similarly. And it's not ideal to switch between our versions in RStudio for every submission, and that's where the RenV package is part of that solution. The recommendations going forward are to add a question or information in your pre-submission briefing book to explain your R-based submission strategy, whether that's fully R or a hybrid type submission that we've done here. Using CRAN or any other curated repo for your sourcing of your packages, using standard packages to minimize any dependencies on sponsor-developed packages, and then that the required packages are installed using a single package list file, and the revloc file is created, and then provide thorough documentation and comments in your ADRG to help with the reviewer. So we've also added a slide here with links to all of the references. There are additional presentations that speak to the package validation process within J&J, and also a more holistic look at our open source journey in R. And we've also linked here the R Consortium pilot projects, which we found super helpful in our preparation of these submission packages. So at this point, I think we could probably start going through some of these questions.
Thank you so much, Alicia, Stephen, and Linshan. This is really, really informative. And very nice to see that you already got approval for one of the submission and then positive feedback for the second submission. And then I see a couple of questions coming in, so I'll start to ask them. And for the audience, please feel free to provide more questions in the Q&A panel. So the first question is about this like SAS versus R hybrid submission. So the question is that I remember hearing from FDA a few years ago that their preference was to receive submission of data packages in one language. And have you heard any feedback from FDA on this hybrid submission approach?
Yeah, we didn't hear anything. So we had explained, as I said, in the pre-submission briefing book, that they would be receiving SAS data sets and XPT, associated SAS code in support of those data sets in SAS, and then output, so table listing figure code is our scripts. And for both submissions that did not generate any type of a specific comment from FDA mentioning that they preferred only one language in the submission.
Yeah, from the R Consortium working group, I also get the impression that from FDA reviewers, as long as you're not using two languages for, say, TLG or two languages for ADAM, that's not a big issue. Like, I think the main reason is they don't want to switch between two languages when reviewing, say, TLGs. Yeah. And then a follow-on question also about this hybrid submission. Do you plan to use R to create ADAM data set in the near future? And if yes, would you use Admiral packages?
So that is one of the things as a company, Johnson & Johnson is looking into, whether or not we would delve into also using R, specifically the Admiral package. There is some piloting going on internally, and there has not been an official decision one way or the other, but it is certainly on our radar as a path forward.
Cool. And then a question about a proprietary package. Do you guys have any kind of like not open source packages used in this submission? Doesn't sound like you have, right?
No, based off of the Novo Nordisk presentation that they had given, I guess now that was a couple years ago, where FDA had came back and said that they didn't want proprietary packages as a part of the submission and had given the feedback that they needed to redo their analysis using open source. So we took that to heart. And both of these submissions and our strategy for all submissions moving forward is to have open source packages and not send proprietary packages.
That's awesome. Yeah. And maybe just one piece of information for everybody that like, although like I think deeply in my mind, I also hope everybody open source your package. But if there is a situation that you need to have a proprietary R package with the collaboration between our consortium and FDA, in the latest technical specification guidance of ESAB, actually one is allowed to submit zip file like a 4R package. So if you have a proprietary R package, you can submit that through the ESAB gateway using a zip file. And then the next question is there a good resource available beyond the env setup Vignette that can help someone build the config file and set up their .R profile in a more automated fashion?
I think that's a difficult question because yeah, we do have the website available and it has very specific examples as well as I have explained with the development area, QA environment and production environment. You have to really go through the entire website to find examples and code and be very patient because it's quite complicated to set up.
Thank you. And I see there are several questions about validation, so I'll try to combine them. The first one is, do you have any issue during validation because of different software being used? Do you do any rounding on statistical results? I think this one is probably more around R versus SAS comparison. And then another question around validation is, could you give an example of how you validate one R package?
So maybe I could start, or I don't know, Linshan, if you wanted to take it, or I could.
Yeah, I can answer the question about how did you do any rounding on statistical result. I think this depends on you working on table or display the value in the figure. So when we display the figures, we will not do any rounding. So one thing, but if we display those values in table, we do rounding, but we have the function called round SAS. That function can round it data in the same way as handled in the SAS. So we don't see the difference for the because of rounding.
Yeah, and I think especially for the outputs, one of the things that we've done just as a pressure test is when we have production code producing outputs in R, for a while, we were also using SAS as the QC, so the check of all of those numbers. And we did find slight differences in, certain statistical models, certain types of rounding, and there is a function that we've included in here to specifically have the round SAS to help match up with what we are typically used to with the SAS type rounding. But that didn't cause any issues in the validation. In fact, it really just cemented that majority of what we're producing, it doesn't matter whether you were using SAS or you were using R, we were getting exactly the same results. And then maybe just to then speak to the additional question around validating the R package, and I'm not going to do this any justice. So there is a link and a presentation that's out there from the J&J team that is responsible for the package validation. And I would just highly recommend that if that's what you're interested in, go take a look at that presentation. It goes into a lot of detail on how we validate the packages internally.
Awesome. Yeah. And then I see there are several questions about kind of like what are like how reviewers review your submission. The first question is like what R system does the reviewer use? Maybe this is more to Stephen and Alicia, like when you have the live session, informal live session with the reviewer, kind of what they show to you on their practice. Yeah.
Yeah, they are working on a Windows environment. So since we're working on a Linux environment, that sometimes poses some issues with package installation, but I think we successfully managed those challenges during the submission.
Well, cool. And then there's another question more about FDA reviewers' activity. I'm not quite sure whether you can speak to it. It says, did you experience any FDA IR requiring rerunning the R script to get a reported result later in time? And if yes, how did you deal with versioning of R within J&J?
I don't think we had an IR on that, but we do make sure that we keep the same version of R within our container that we work in, so that this issue doesn't appear.
Well, sounds good. And another question, maybe also a little bit related to our package validation and package quality assurance. How to assure quality of the packages from CRAN. And I assume you probably have a kind of like a preferred list of packages, but I wonder whether you can share a little bit more on that. Yeah.
Yeah, I think so Stephen kind of alluded, right? We have a container internally that will give us access in a production environment to only those open source versions that we've internally validated or verified and are comfortable using. And as mentioned, that's also explained in that previous talk given by J&J folks on package verification within J&J.
Awesome. Awesome. I see like there are questions about whether you will be sharing your presentation and also the link to the J&J presentation. Maybe we can make sure those are shared this event. Yeah.
Absolutely.
Yeah. Cool. And one question about this more like a people side, kind of like the R adoption. How did you facilitate the adoption of R packages among SAS programmers?
That's $1 million question. And something that I think we're still working through here at J&J, and I'm sure others are as well, right? But there is a strong shift that we see in the industry with an adoption to R and of R. Our statistical counterparts are also using our counterparts in other functions here at J&J are using R. So it's a natural progression forward for our statistical programming team to adopt R as well.
Thank you very much. Cool. Yeah, I think we're going through all the questions. I do have 2 questions from my side, so maybe I'll ask from my side. And for the audience, if you have more questions, please feel free to add more into the Q&A channel. So from my side, one thing I'm curious about is I see that in your submission 1, submission 2, basically for the first submission, the FDA reviewer asked for this mock submission, and in the second one, they didn't ask for mock submission, and it seems like they are pretty comfortable with R-based submission. I assume those are different individuals. So what you see is maybe like individual preference from the reviewer side. Yeah.
I think I do think there was an individual preference. You're probably, 100% correct on that. I also think too, it's an ask, right, of FDA, because we're sending them an additional package. We're asking them to take their time away to review that package and provide us feedback back. So we didn't expect that we would have that. Necessarily that we would have that 30-minute informal meeting with them, but given the feedback back and the struggle that they were having, we thought that it was best moving forward that we could just have a quick kind of face-to-face, almost, virtual face-to-face, and just go through the process. I do believe that was really dependent on the reviewer themselves, right? So that they felt more comfortable kind of going through it, knowing that they wouldn't get stuck, have to send an IR, wait for that response, and then potentially another IR, wait for that response. And from the sponsor side, so from J&J side, we also wanted to minimize that back and forth happening when, you're already in a time crunch because they're reviewing your submission. There's a PDUFA date, there's rest of world submissions that go out and you're getting other IRs. So I do think from both sides, the J&J side and then from FDA side, it was very helpful for them to have, to accept that idea of a mock and then go through it. And then I think it helped with how smooth the submissions went moving forward.
I think that's an amazing story. I feel like kind of like we are all going through this transformation together from industry side and also from the health authority side, and then different people at different stage. And I think this is a wonderful story to remind people that collaboration is really important during this period, and there is a human like behind the system, and we really should talk and then to make it as smooth as possible. Yeah. Yeah. And then my last question, like more wiring my head from our submission working group. And I know you referred to the submission working group pilot. I'm really happy that those material has been useful for your real submission at J&J. Maybe just like want to ask for any feedbacks or ideas for future pilot. What are the gaps you saw between the public available pilots versus what you guys need to do in a real submission?
It's a great question. I don't know, Stephen, Lynn, Sean, if you have any thoughts.
Yeah, my concern is about the number of the package, the container. Because we keep update the container and the number of the package is growing. So now you could say maybe it's 100 packages, but later on could be 200, 300, how we figure out a better way, only only include or extract the package just typically used for the study, the particular study, rather than send off the entire, most of the actually won't use in the involved in the particular analysis of submission. Yeah, so that's my concerns. I was kind of interesting. So what's the possible solution in the future?
That is a really practical one. I think we all start from a selected number of packages, but we're not using every single package in a submission. Yeah. Anything else from you, Alicia and Stephen? Not much, yeah.
Thank you.
Well,
Yeah,
our consortium is doing such a good job. I mean, they're really covering all the bases, I feel so.
Thank you. I'm really glad to hear that. And I'm very excited about the latest pilots of using container and also of using JSON.
Yeah, me as well. Yeah.
Cool. With that, yeah, thank you so much for the very clear and very informative presentation. I'll turn it back to Jesse to do the closeout.
Yeah, just I think we'll conclude the webinar here. I want to thank participants for joining and listening in and asking questions. Our consortium's mission is to provide these kinds of platforms with collaboration and information sharing. Big thanks to Linshan, Stephen, and Alicia. This is a level of depth that's just fantastic, and it's great to see the progress that you've made. It's hard work. It takes ongoing effort, and it's just great to see this. So thank you to our three speakers. And we'll finish here. Thank you very much, everyone.
Thank you.
Bye-bye.
Thank you. Bye.